Well, thank you, Jenny. Ladies and gentlemen, welcome to NHRI Forum Learning Health System Workshop on SARS-CoV-2 and COVID-19. The first session of this workshop will be given by Professor Michael Lai Mingzhao 教授. Dr. Lai has his MD from National Taiwan University and a Ph.D. from UC Berkeley. He is a member of Academy and currently he is Distinguished Visiting Chair of Institute of Molecular Biology of Academia Sinica. He is also an honorary professor of many universities such as University of Southern California, National Cheng Kung University, etc. He is a well-known virologist in the world and had made significant contribution in the hepatitis viruses and the coronaviruses. Enlightened by Professor Leslie Weiner, Michael started his coronavirus research when he was a young assistant professor at University of Southern California. Very few people studied coronavirus at that time before the SARS infection. Well, we all know that is because people thought coronavirus does not give serious diseases, and therefore it's difficult to get grant. But Michael persistently kept on his research on coronaviruses and paved up the groundwork of understanding this probably the most important virus today. Many of our virologist colleagues call him the father of coronavirus. So it is really our great honor to have Dr. Lai Mingzhao to give the leading lecture of this workshop. The title of his talk is The Challenge of Lassoing COVID-19. Michael, please.
Thank you for the opportunity to share with you my perspectives of COVID-19. First of all, I'd like to explain to you why I use the title, The Challenges of Lassoing COVID-19. Lasso is a device used by American cowboys to train and to tame the wild horses so that they can be ridden by human beings. Lasso would not kill the horse, but would tame the horse so that they can be ridden by human beings. So that reminds me very much the stories of COVID-19. I think COVID-19 can be controlled, but cannot be eradicated. So that we learn how to work with COVID-19, just like we work with wild horses. So I think lasso is very appropriate for the controls of COVID-19. Okay, COVID-19, we can understand COVID-19 from several different aspects. The first aspect is molecular virology. I said in my talk, I will present the perspective, my stories from the perspectives of virologists, but I'm going to present a lot of scientific data. The first aspect of COVID-19 should start with molecular virology. The studies of viruses usually start with the studies of structural proteins. The coronavirus, the spike protein, was known in early 1970 when there were few studies on the virus, coronaviruses. The most important structural proteins, of course, is spike protein. Both spike protein is the vehicle for virus infection and also a target for immune protection, because this is the very important protein, and we know a lot about spike protein. But in addition to spike proteins, we should not forget that the M protein is also virulent protein, nucleocapsid protein. But in addition to that, it also has regulatory roles, as shown recently by Ye Shufei 博士. She demonstrate that N protein can regulate mRNA transcription. In addition to S protein, N protein, two smaller proteins which are usually very often neglected are M protein and E protein. M protein, E protein can affect virus maturation. And we know that M and E proteins are not always necessary for virion particle formation, but the inclusion of M and E protein makes the virus particles more compact and more infectious, as recently shown by Yi of Cheng Kung University. So I think this is very significant. As you all know, that one characteristic of COVID-19, this SARS-CoV-2 COVID-19, is that the infectious virus particle production does not correlate with the symptoms. So this means that the release of the virus particles are not rigorously controlled by the virus particles, by the S protein, by the S protein alone. So the M and E proteins may play a very important role in controlling virus particle formation. And there is a crucial point of the COVID-19, because COVID-19 the virus production and the symptoms do not correlate. So it's difficult to control the production of the infectious virus particle. So I think this is one aspect which should be studied in the future. Now, the non-structured proteins is coronavirus is very unusual. The coronavirus, the non-structured proteins constitutes almost 2/3 of viral genome. So it is very unusual to have a virus, particularly RNA virus, to devote so much gene space for non-structured proteins. There's 16 non-structured proteins just simply for replication of the virus. So these non-structured proteins very often, usually are provided by the host cells, but for coronavirus, it deliberately devote its own gene space to make this protein. That's unusual. We don't understand why it needs so many proteins. So this is one aspect we should think about it. Why it needs so many non-structured proteins? Why does the virus bother to make so many proteins? So that when the sequence of coronavirus, first IBV, infectious bronchitis virus, the mouse hepatitis virus, the sequence was known from 28 kb to 32 kb. It was found that the non-structured proteins are made from one big protein, the protein 1A, 1B. And these proteins all devoted to virus replication. And so many proteins, so many unknown proteins, and the identification of these non-structured proteins was done by brilliant bioinformatics scientist Alex Gobardenian of Moscow, which this will set the foundation for functional studies of these proteins. So I think this is a very important area of work. The structure of most of these proteins now is known, which opens up the potential avenues for antiviral research. So I think the structured proteins and non-structured proteins will be a very fruitful area for research. Let's turn to the RNA. The RNA has a 32 kb RNA. This is the biggest RNA among all the RNA viruses. And it has made the seven subgenome messenger RNAs. Messenger RNAs are nested from 3' end and jumping to the 5' leader. And subgenomic messenger RNAs are made by discontinuous transcription. Now everybody knows that. But replication of genome RNA is made by continuous RNA synthesis. So you can see The transcription and replication are made by two different mechanisms. One is continuous, the other is discontinuous. So keep that in mind, that the continuous transcription and discontinuous transcription are controlled by different mechanisms. So they are probably involved in different proteins. And so you can see some polymers, the transcription and the replications probably involves the same polymerase, but different viral and cellular factors. So replication is the mechanism of replication actually is different from mechanism of transcription. So replication and transcription involves different cellular factors. So keep this in mind. So the replication is not simply transcription twice from positive to negative to positive. So they are not mediated by the same mechanism. They use different factors. Subgenomic RNA. So this is one thing which should be kept in mind. I saw recent studies. Some people claim that the replication and transcription are the same. So they use the same, involve the same kinetics and so on. So I think that is not. I think it's not correct. So keep that in mind. There's replication mechanism and transcription mechanism are different. Another point is the subgenome messenger RNA are wasteful messenger RNAs. Why do I say that? Because only the 5' open reading frames are translated. So the remaining messenger RNAs are wasted. So why the virus want to waste so much RNA? So I think there must be some reason for it, because many viruses use this subgenomic messenger RNA mechanism for their protein translation. So there must be reason to preserve so much messenger RNA space. So my thinking is that probably the messenger RNA sequence following the first open reading frames actually may play some roles that plays the regulatory functions. So may play regulatory roles in this part, the 3' of the messenger RNAs, which may play some regulatory roles in transcription or some other functions. So indeed, we have some data that my former student Helen Liu, she showed that for some positive RNA viruses, indeed the transcription and translation are linked in some linked. So they may play a role in transcription as well as translation. So this is one area which we can further study in the future. Okay, now let me switch to another topic, the viral instability and the variants. Now we all heard our story that how coronavirus mutate, the new variants appear all the time. So that means the coronavirus RNA mutated very quickly. These mutations or the viral mutants arise through errors in replication, balanced by environmental selection. So that is the mechanism for appearance of the mutants. The RNA virus, therefore, RNA virus are quasi-species consisting of multiple genomic varieties. Why coronavirus RNA undergo such frequent mutation? And one reason is that the RNA polymers of RNA viruses are inherently error-prone. The replication is not very precise. Therefore, during replication, they make a lot of mistakes. So RNA polymers, the mutation frequency, error frequency is much higher than DNA polymers. But among all the RNA polymers, the mutation frequencies can be as high as 10,000-fold, 1,000-fold, or 10,000-fold, or even higher than the mutation frequency of DNA polymerases. But among other RNA polymerases, the SARS-CoV-2 RNA polymerase mutation error frequencies actually is not that high. Although it's higher than poliovirus, but it's actually is lower than influenza virus, or of course even lower than HIV. So the RNA polymerase of coronavirus has high mutation frequency, error frequency, but actually is not as high as some other RNA viruses. Nevertheless, yes, it can cause a lot of mutations during replication. Another factor coronavirus has higher mutation frequency, very often mutate, is a genome size. The bigger the genome size, the higher probability of accumulating mutations. Coronavirus has a 31 kb RNA genome, the longest among all RNA viruses. Therefore, they tend to make a lot of mistakes accumulate a lot of mistakes during replication. On the other hand, SARS virus has some other points, some other mechanism to keep the mutation frequencies down. Like SARS virus has a RNA repair enzyme. There's a non-structured protein 14, there's a 3' to 5' exonuclease. And that is the only RNA repair enzyme known. So that makes they can repair some of the mistakes, remove some of the mistakes to keep the mutation frequencies down. Another mechanism is RNA recombination. RNA recombination is very often allow the viral RNA to repair the mistakes, because then it can jump from one RNA to a different RNA. If the different RNA contains correct sequence, then the mistakes in one RNA strand can be repaired by the second RNA strand. Therefore, the mutation frequency will be kept down. So this mechanism allows the mutation frequencies to be within the manageable level, manageable range. But once the mutation is made, how are the mutants selected? The thing is due to the selection pressure. The mutant RNA, mutant virus can replicate very well, can have the advantage over the wild type RNA. Then this mutant RNA will appear. What kind of selection advantage the virus might have? They might have high transmissibility, high replication ability. They'll have ability to escape from immunity, the natural immunity, or the vaccine-induced immunity. If the mutant RNA, mutant virus has this ability, then the virus will replicate, will become the dominant virus. So this is how the mutant virus is selected. Another point is that some mutant virus might have ability to cause more severe disease, such as the hospitalization rate of the patients, the age of patients. And further, these parameters have any correlation with the ability of the mutant to survive. That is not certain. It's not certain. And in particular, like now we heard recent COVID-19 mutants with Delta strain. Delta strain now they cause more severe disease. And so whether the severe disease severity have any relation with the selection advantage, that is not clear. But although the SARS-CoV-19 now it seems that becomes more virulent, but I feel that this is SARS-CoV-19 is still at the early stage. SARS-CoV-2 is still in the early stage of virus evolution. So I feel that when the outbreak, this pandemic continues, the virus will tend to lose the the toxicity, the potency. So the trend of the virus evolution is to become less virulent to humans gradually. So with that discussion, let's look at the real data. This is a famous paper. You can see what I'm showing here is the orange virus. Orange virus starting from January 11 to March also. You can see that in these 3 months, almost all the viruses are orange color. Now, starting February, blue viruses started to appear. Now, up in May or June, almost 100% of the viruses are blue virus now. Orange virus and blue virus, the difference is only one amino acid in the spike proteins. So you can see one amino acid change now allows the blue virus to take over the virus populations. The orange virus disappeared and blue virus completely. By June, 100% of the virus of the COVID-19 in the world were blue virus. Then there is one single amino acid mutation. So you can see this is the virus evolution. How one single amino acid changes will allow the virus to have higher transmission rate and then take over the populations. Now, starting in a few months later, now the new strains of virus appear. This virus is the now starting October of 2021. Now the new mutants appear. In UK, alpha strain appeared. Then South Africa, beta strain appeared. In Brazil, gamma strain appeared. In India, delta strain appeared. All these viruses have higher transmissibility, particularly the delta strain. The delta strain has high transmissibility higher than alpha strain, and of course higher than the original virus, blue virus I just showed you, and the high hospitalization rate, younger age of patient, and also escape from some vaccine-induced immunity or the natural immunity. So Delta virus becomes the dominant virus. Now Delta virus is almost 100% of the new infections in the world caused by Delta virus. So this kind of variants continue to increase. Alpha, beta, gamma, delta are considered to be variants of concern, means they have more significance, importance to human beings. But the rest of strains are less important, but potentially they may become very important. So that is something we need to worry about. That is because the properties of the virus can mutate very easily, very quickly. So that reminds me one point, because RNA, virus RNA is a quasi-species. So that means they have accumulate a lot of different RNA sequences, different RNA. Then these populations of the viral RNA are selected out by environment. Certain environment will favor certain kind of mutants. As a result, it select out a certain mutants. So that reminds me a question very often people ask me. He said, if two people infected with two viruses of similar sequence, are they infected by the same virus? Can you say that they are infected by the same virus? If the two individuals infected with virus of different sequences, they have different sequences, can you say that they are infected by different viruses? So the same virus sequence, can you say that they are infected, they have the same virus origin? The different virus, can you say that they are infected by different viruses? The answer is half right, half wrong. If they are infected by the same sequence, virus of the same sequence, you can say they have the same origin of the infection. But if they infected with different virus sequences, then you cannot say that for sure that they are infected by different viruses, because virus sequences are determined by selection. Different individuals, even if they infected with the same viruses, they can come up with different viruses. So the same virus origin can result in different virus sequence in individuals. So there is something you need to think about. I often, from TV report, I often heard this mistake. The third issue is, of course, vaccine, which is a very important issue. Vaccine development signifies a historically giant leap forward in terms of speed of development and novelty of conceptual and technical design, which coincided with coming of age of the messenger RNA technology. For the first time in history, there are so many competing vaccines for a single disease. So really the golden age, we can say that we are now in the golden age of vaccine development. So now there are 7 WHO approved vaccines. And they are categorized in 3 different categories. One is the messenger RNA type, and Pfizer, BioNTech, Moderna. Everybody now heard so much about these vaccines. Now the second type, AstraZeneca adenovirus, Serum Institute in India. So they're all carried by adenovirus. And also the Johnson also adenovirus. And then the third type is the Chinese vaccines. These are whole inactivated coronavirus. In addition to these 7 approved vaccines, there were over 20, 30 different vaccines being approved, being studied by WHO. So very soon we'll have a lot of different vaccines. So this area will be covered by the next speakers. So I'm not going to devote into the discussion of this vaccine. But I just want to point out that this vaccine development is so new, so exciting, that there are many new issues which we need to think about it. For instance, the vaccines are effective against various different types of variants, particularly the Delta variant. The adverse reaction to vaccines is a very significant. It's a very big issue. It's a vaccine advantage over disadvantage. So there's something we need to think about it. How many percent population vaccinated is considered to have reached herd immunity? Whether we need to have booster shot, whether we use a different type of vaccine, a mixed vaccine, hybrid vaccine, whether it's an advantage. This vaccine speed up generation of variants because we do vaccination, so the variants appear more quickly. Is that true? And breakthrough infection. Why people who have been vaccinated also could develop, could be infected again. And then the social issue, vaccine hesitancy. There's some people would not vaccinated, will not be vaccinated because of the cultural reason. How do we overcome that? And of course, the emergency use authorization is okay. So these are many issues I think that will be covered by the next speakers. So I'm not going to details of this. Another issue is the pathogenesis. Why the virus develop disease? The most important reason is that the receptors for the virus is ACE2. ACE2 activated by the virus that allow the virus to undergo endocytosis and get into the cells. So the ACE2 is very important for virus entry by infection of the cells. But ACE2 binding by virus by itself actually activate ACE2 cascade. the ACE2 now can activate another signal transduction. It activates the renin the ACE2 pathways, and that will activate another signal transduction, involve the steroid hormone and so on. So this ACE2 cascade is by itself, and by activating by virus will cause another symptoms. And the virus with ACE2 infected by cells with ACE2 infected by virus now will cause the disruption of the cells, and will cause the symptoms. There's symptoms in respiratory tract, the immune mechanism, and also virus can bind to other proteins other than ACE2, and that also causes disease. So all this mechanism together will contribute to the disease. I think this will be covered by the next speakers as well. So I'm not going into detail. You just want you to remember that the disease itself is not only the virus infection of the cells, but you activate ACE2, activate renin ACE2 pathways. You also activate immune mechanism, and also activate other cellular factors and so on. And the virus can also infect other animals, such as mink, raccoon, and so on. And whether it can infect other domestic animals, there is no other coronavirus are known to cause cross-species infection. So all this contribute to the disease caused by this infection. Well, other disease symptoms recently reported, brain symptoms. The patients who have memory loss, who have cognitive disorders. And this is very similar to murine coronavirus. Murine coronavirus can also infect the autoimmune coronavirus. The primary target is liver cells, liver hepatocytes, but it also can infect, have neurotropism, can infect CNS infection. But its target is oligodendrocytes. It causes myelin loss. So they are different. It is the range of infections is similar between murine and COVID-19. It also can cause a long COVID-19. Some patients have prolonged disease after 8 months, 9 months. The symptoms still persist. And this is very similar to murine coronavirus. Murine coronavirus can also cause persistent infection, like not only has a persistent infection of brain and so on. So that is very similar to the COVID-19 and the animal coronavirus. So the mechanism might be different, but might be same. I just want you to keep in mind that coronavirus and that ability. The final issue, I just devote a few minutes to this issue, the origin of SARS-CoV-2, because this recently there are a lot of arguments, like the White House, United States White House, U.S. Congress, and WHO, and many people are interested in this issue. The SARS coronavirus, man-made, or is a natural virus, natural evolution of the virus. We know that SARS coronavirus 1 is the original SARS virus. It's a zoonotic virus jumping from bat through an intermediate host, the palm civets. So there's no question that SARS virus 1 is derived from animals in nature. The SARS coronavirus 2 also has a close relative in bat. This bat has 96% homology with SARS. So you can see that probably is also derived from animal, from bat. So both SARS-1, SARS-2, and also MERS virus are derived from bat, that's for sure. But the SARS-2 has a receptor binding domain, which is significantly different. There is 5 out of 6 critical amino acids involved in the binding to the receptors are not related to bat virus. So they have to be derived from somewhere else. So where does this RBD domain come from? And so we don't know. The answer is we don't know. It will probably been made or it probably from somewhere else. Now recently it had been shown that 2 more bad viruses have been found, and they have even higher similarity with SARS-CoV-2. So the question is that this actually is derived from some other bad virus that is not sure. The SARS-2 binds to ACE2 significantly more strongly than SARS-1 or bad virus. So it seems that if it's derived from, if this, the receptor binding domain of SARS-2 is derived from nature, usually is the jump, the increase in the binding affinity just slightly. It's not like the SARS-2 here. The jumping is from 1 amino acid to 6 amino acids. So there's a big jump. It's not like natural virus evolution. So because of this, people suggest that it might be different from, it could be a man-made origin. And I think that the most persuasive, convincing evidence to me is that this COVID-19, the RBD sequence, this new receptor binding domain sequence, its codon usage patterns is different from that in other genetic region of the virus. So the virus has its own coding sequence pattern, but the virus in the RBD, in the COVID-2, the coding usage pattern is quite different. So that's why people suggest that this could be man-made origin. But I should say that this is really all speculation. There's no hard evidence. And also, the serological surveys appear to show that no evidence of viral antibodies in workers or customers associated with the epicenter of the outbreaks. So this is just show you that the blue is the sequence related to that virus, but the pink is a region related to unique to SARS coronavirus. So I just too much data. I just want to show you that this RBD domain, the sequence, is different from the remaining sequence, which is similar to bat virus. So most of the SARS-2 is similar to bat virus, but a small region is similar to something else, which we don't know. So at the moment, all we can say that this is a tale of Rashomon, Roseman. That means you can gongso gongyuri, also really. That you just use your imagination to say that where does this RBD come from? So finally, we are in a very, I feel that we are in a very tumultuous and uncertain period of time. Science is very best. And you can see it's amazing that just in a year or two, less than 2 years, there's so much progress in science. But the virus is also at the most elusive. The vaccine gives us unprecedented power to combat the virus, but the virus countered with uncanny ability to elude the human efforts. This difficulty is exacerbated by the divide between the rich and poor countries in the world. I cannot imagine we will ever eradicate the virus. We will only lasso it and coexist with it. So we hurry the scientists and medical personnel for the efforts. Thank you.